dear children, let's begin with the first meiosis, okay, meiosis 1, in which it begins with prophase 1. So it is the longest and the most complex process which is occurring in meiosis. And again, the prophase 1 is subdivided into 5 substages. They are called as leptotin, zygotin, pachytene, diplotin, and diakinesis. So 5 stages in meiosis are, I repeat, leptotin, zygotin, pachytene, diplotin, and diakinesis. So these are the 5 stages. Let us learn in detail. So the longest phase begins with leptotin, in which the chromatin fibers, which is formed, which is seen during the interphase stage, it starts to condense and form or take the structure of long, slender chromosomes. So throughout the prophase, the condensation of chromosomes takes place and the nucleus enlarges because a large number of activities is going to take place within the nucleus during the prophase 1. So leptotin begins with condensation of the chromatin fibers to form the chromosomes and the nucleus enlarges. The leptotin stage is followed by zygotin. In zygotin, the process of condensation of chromosomes continue, and as I told you, in diploid organisms, 2 sets, one from the paternal and one from the maternal parent, is present. And of these chromosomes, 46 chromosomes, the similar chromosomes means in terms of size, shape, and also the genes they carry also will be the same. So such similar chromosomes will start to pair with each other. And this pairing of similar chromosomes is called as synapses. And the synapses of the similar chromosomes is helped or aided by synaptonemal complex. So this is the pairing up of similar chromosomes. And such paired chromosomes are called as homologous chromosome. So this pair, which is constituted by red and blue chromosome, is called as a homologous chromosome. And each pair of a homologous chromosome is called as a bivalent. So this pair of a homologous chromosome is called as a bivalent. The zygotin is followed by a longer stage which is called as the pachytene. So what pachytene means is the stage in which the bivalent chromosomes in a pair, they split apart. Okay, they split into, see, each chromosome, they split into similar chromatids. So here we can see the blue is having the blue with its sister chromatids and red chromosome with its sister chromatids. So such a bivalent with the split up chromatids is called as a tetrad. Okay, and appearance of recombination nodules occurs at which the crossing over takes place. Okay, the crossing over takes place. I will tell you what is crossing over. So in order for crossing over to take place, recombination nodules appears. Okay, and it leads to recombination of genetic material on the homologous chromosome. So this pair is called as a homologous chromosome. And as I told you, each chromosome having its own sister chromatids. And when there is an exchange of genetic material between the non-sister chromatids, here the arms of the blue chromosome and the red chromosome can be referred to as non-sister chromatids. So non-sister chromatids, between the non-sister chromatids, if there is exchange of genetic material taking place, non-sister chromatids, genetic material is getting exchanged in a homologous pair, this process is called as crossing over. And for crossing over to take place, recombination nodules are formed. So recombination is completed by the end of pachytene. So here we can see at the position of this recombination nodules, exchange of genetic material is taking place. So the pachytene is followed by diplotin. So diplotin, what happens? The synaptonemal complex which helps in the pairing of the homologous chromosomes dissolves. Okay, and the recombined homologous chromosomes of the bivalents, they separate from each other. Okay, so these homologous chromosomes which were paired together, and this site of pairing can be considered as a synapsis, or where synapses occurred, there was a synaptonemal complex. And due to the dissolution of the synaptonemal complex, the chromosome starts to separate from each other, but they remain attached between the non-sister chromatids at the site where crossing over occurred. Okay, and this X-shaped structure is called as the chiasmata. So this X-shaped structure is called as a chiasmata, and it is this place where the recombination of crossing over occurred, and this is followed by diakinesis. That is the terminalization of the chiasmata occurs. It moves towards the tip. That's called as terminalization. So chromosomes are not fully undergo complete condensation by the time they reach the last stage of prophase 1, that is diakinesis. And the meiotic spindle, okay, the meiotic spindle, they start to originate from the poles. As I told in mitosis, the centrosomes, they move towards the opposite poles, and from the centrosomes the spindle fibers start to appear, and the different structures like nucleus and nuclear membrane starts to disappear, and thus all the chromosomes remain scattered in the cytoplasm. So spindle formation is completed. You can see the spindle formation is completed, which is originating from the poles, and the chromosomes in the metaphase 1. Okay, prophase is followed by metaphase 1. They start to align along the metaphase plate or the equatorial plate. And the microtubules of the spindle apparatus, they come and attach to the homologous chromosomes. They come and attach to the homologous chromosomes. We can see these are the homologous chromosomes. Okay, so they start to attach at the kinetochore constriction present on the centromere, and this is followed by anaphase. And what happens during anaphase? Contraction of the spindle fibers occurs, and in the case of mitosis, it was the sister chromatids who separated, whereas here, what has separates? This is a homologous chromosome. A pair of chromosome similar together is called as homologous chromosome. So the homologous chromosome, they separate from each other. Okay, while this chromosome is having its own sister chromatids, and this is having its own chromatids, and the chromosome, the separated chromosomes of the homologous pair, move towards the pole along with its sister chromatids. Is it clear? And this is a metaphase in which they separate, and this is anaphase in which the homologous chromosomes separate from each other. And this is followed by telophase. The nuclear membrane and the nucleolus reappear, and two haploid daughter nuclei are formed. We can see two haploid daughter nuclei are formed, and this is called as a dyad. This is a dyad. So these were a homologous pair. Imagine this was a homologous pair and this was another homologous pair. After this, the cytokinesis may or may not occur. And after a very short interphase, it is followed by meiosis 2. And the stage between two consecutive meiosis, that is meiosis 1 and meiosis 2, is called as interkinesis. And DNA replication does not occur during this interkinesis. DNA replication had occurred only in the first interphase, which is occurring before the prophase 1. So dear children, just in this module, we have come to an end of the longest phase in meiosis 1, that is prophase 1, which is subdivided into 5 processes or 5 steps, which can be called as leptotin, zygotine, pachytine, diplotin, and diakinesis. And all through these stages, the condensation of chromosomes take place. Pairing of homologous chromosomes takes place. Pairing of chromosomes is called as synapses, and during the pachytine stage, crossing over takes place between the non-sister chromatids, and then during the leptotin stage, the chiasmata is visible due to the separation or dissolution of the synaptonymal complex, and during diplotin terminalization takes place, and during metaphase, all the homologous chromosomes, they come and align along the metaphase plate, and during anaphase, the homologous chromosomes separate and move towards the pole, and during telophase, the completion of the meiosis 1 karyokinesis takes place, and this can be followed by cytokinesis or not, and it results in a dyad. Thank you.